Thank you for the invite. Thank you everyone that stayed throughout for this last session. I hope to make it interesting for you. I plan to focus on the concept rather than products, and I hope this is thought-provoking. I'd actually like to get this started. My group is focused on the interface between AI and microscopy here at Leica. This is the group. I wanted to give them a shout out straight away. I don't know, do you see this on the top here, or does it not show? All the Zoom things seem to be covering most of my...
It's hidden.
We can see your complete slide, thanks.
Ah, awesome. Okay, very good. So I wanted to introduce the team that is responsible for a lot of the work, actually for all of the work that is presented on the slides after this. It's a large team based both in Bellevue and near Seattle on the Pacific Northwest in the US, as well as in Germany, and then a little bit spread out all over the world. It's really a wonderful team to work with. I feel, yeah, honored and very lucky. This is some of the work that they've done and we've done and released over the years. The history here is really for Avia. But as you know, Leica has a lot other software. Avia itself was part of S-Vision, and I was there starting the project there with Avia 5. And Avia is for rendering, displaying super large data sets and doing analysis. There's also cloud computing. There's segmentation of various types of structures and tracking. So it covers a broad range of applications. The real distinctive feature is about using AI for a lot of the more challenging tasks, both on segmentation and classification or phenotyping, as you might wish. But I will park it there. I didn't really want to talk about the product for the whole time. I really wanted to talk about a concept that we started to explore about 2 years ago. And now we feel pretty excited about. So as I mentioned, this will not be a product introduction, or it won't include any product demo either. It's not going to be a technical lecture about deep learning either. And I really want to think with you about the concept that is presented here. So let's get to it, and I'll show some results as well, to show that the concept is not just in my head, in our head, but it's possible to implement it and get some interesting results. Because this is all going to be based on machine learning concepts, I have a couple of slides just to get everybody on the same page. Perhaps you're a machine learning expert, and this will be your time to go get some water or a coffee. You get about 2.5 minutes or 3 minutes, and then you can come back for the rest. But these gentlemen are credited with the concept and the coining of the term machine learning. That's Alan Turing, and then Arthur Samuel for the first implementation that could actually be tested. That was only the beginning, and there were many AI winters, several, actually 2. And you've probably heard about this. More recently, the last 10 years or so, especially deep learning has become really popular, mostly because compute power is cheap in comparison to previous times, and there is a lot more annotated data, so it can be really, you can use those concepts, some of them from the 80s, like Jeff Hinton with artificial neural networks. That concept has been around for a while, but just very difficult to use. And also convolutional neural networks, which is really at the core of pretty much all neural networks that we use today, are from the late 80s with Jan LeCun. These gentlemen are now very famous, of course. They've won several awards in recent years, but their main discoveries are really a few years, decades even old. But they couldn't be implemented at scale until the last 10 years. I called out here Ian Goodfellow because I think this kind of technology, the adversarial neural network, has a special place and really has the potential to create new universes. Really, I'm not going to go into the details here, but it's very exciting. That's why I'm calling him out early, even though he hasn't won awards as much as Jeff and Jan LeCun. And Olaf, you probably know him. Maybe some of you even know him personally, but he's become super famous in the last 5-6 years with his UNet, which is probably what Tom and the others use in their, yeah, it's so robust. We also use it, yeah, it's great technology, good basis to start build your own stack. But these are just a few. There's actually quite a lot of other people that contributed, and I only listed a few even of the ones that contributed. If you want to know more, this is one of the best papers I found that do a good overview here by Jürgen. This is called, in neural networks, and you can find it. It's from 2015. It covers really a lot of the details of the historicals and the key contributions over the decades to get to where we are. Some cool developments, cool from my point of view. So I call these out because I find them exciting, and they were enabled by deep learning. Some of them are kind of just for fun, but more recently, 2017, you start to see things start to come into the life sciences and more specifically into medicine. And that gets real, that gets serious, because you're now talking about an AI that is able to have a direct impact on human health. And that's very interesting, of course, very close to the heart of all of us here, as we are all in the life science space. So the people at Arteris, Viz AI, and Imogen managed to get FDA approval for their applications. There's several more in the meantime. These are for heart disease, stroke, and bone disorders. These are predictive measures, but there's more. And more recently, and I just swapped this out a few days ago, more recently, I'm particularly, my mind is still kind of grappling with this. If you haven't read the interview, I recommend it that you read it. But this particular chatbot claims to be conscious, and it's a long interview. It's probably like a 20-minute interview, 20-minute read of the interview, and it goes back and forth with a couple of Google engineers, and it is impressive how, I don't know what to call it, self-aware the thing might sound. Now, for the more technically minded audience here, you might say, okay, that's just, that's what the technology should look like. That's what it should do. It's just very good at it. But it does make you wonder, make you think about, how far have we already come, and how far are we from making that bigger leap that we don't really know exactly what's going to happen. But it is a chatbot. It is very interesting. I just suggest, if you haven't read the interview, it's pretty interesting, thought-provoking for sure. So huge developments over the last the last decade, really, and they can be grouped in this way. I think many of you have seen this. So artificial intelligence, the big group. Then you have some algorithms in there that learn from data. Generally, they're called machine learning algorithms. And then you have the, let's say, Formula 1 version of machine learning, which is deep learning. It has so many more parameters, has a lot more capability to learn from the data, and especially when there's a lot of data and the groups or the predictions required are more nuanced or sophisticated, then deep learning is really the thing to do. This is a critical one. I got it. You can see here, this is a great blog also, if you want to read up on this. This is a great blog post, and the picture itself is from NVIDIA. And it summarizes the key concepts between the comparisons between traditional programming and machine learning. And this is at the core of what I want to talk about next. In traditional programming, generally the engineer that has to set up the rules programmatically, writing down all the rules, we put some raw data in and we get some results, some segmentation results, for example. Just for the example, you put some algorithms that will do a threshold, you put some data in, and then you get some segmentation maps. With machine learning, typically you have to provide the segmentation maps up front. You have to provide annotations, manual annotations, or somehow predicted annotations. like what Tom showed earlier. You then provide some raw data, and you can get some predicted rules about what the annotation should look like when you provide new raw data. So the deep learning model is really predicting the edge of the objects in the case of segmentation. This is good and it works. So we also do that a lot at Leica. So we have in Avia, we have already for more than 5 years the capability to train segmentation models in Avia with a bunch of annotation tools, and you can deploy it in the cloud, you can deploy it locally, so on. So the user has to provide ground truth annotations, as I said, raw data. We then can train different variety of models. UNet is definitely one of them, but we use a few others that we like. These models can then have the power to create essentially another channel. It's a confidence map channel that will be applied on the raw data, and that confidence map can be then converted to segmentation maps and into 3D, beautiful 3D reconstructions like what was shown in the previous presentation. This is not what I'm talking about. This is not at all the concept that I'm talking about today. But this is when we talk about deep learning and using deep learning for image segmentation, this is the most common way to do it. And it works a lot of the time. However, we thought about something else, and that's what I wanted to talk about. So if you are the AI expert and you just went for coffee, and now is the good time to come back. Or if you're listening in the background, this is a good time to come back to the presentation, because now it should be something different for you. All right, so we're not doing this in what I'm going to talk about next. We do it, and it works for a lot of cases, but in some cases, it's not possible to get the ground truth, for example, at scale. That's one critical example. It's hard, it takes too much time, and simply no one will want to create the ground truth to train these models. So in an example like that, it would be better to do it in a different way. Another thing that got me thinking, was we already have software. There's image analysis software out there for a long time. I think ImageJ, I mean, it's like 20-something years, or Fiji. And this software is used by millions of people. You know, I'm thinking about the whole period of time, all the users. It's probably quite a lot of use sessions. So typically, the images go in, they look a little bit like this. These are fluorescent images of neurons, just happens to be neurons, like the example I have. And you open whichever is your favorite software, you take an image in, and then you will do lots of things to the image. I'm talking about standard image processing, not AI. You do lots of things to the image. You can maybe do a pre-processing step. You might apply some thresholds. You might apply some filtering after for detecting just the objects of the right size. You might do even some associations, and then you have a result. So the result will look a little bit like this. In this case, is our neurons. So we actually have associated the soma to the dendrites to the spines. We have a whole architecture of the objects of interest. We even can get circuits. So all of that is interactive. The user is looking at the GUI, is going through the steps or workflow, and applying a series of algorithms and doing a series of interactions with the image. We estimated that just for the neuroscience space, and this is probably an underestimate, you can see here how I estimated this, something like 300 work years are spent globally by the neuroscience community to trace neurons. And this includes AI and AI helped or aided, AI aided neuron tracing time. It's a lot of time. And all these people will just do it because that's what they need to do for their research. But the concept was this can't be a good way. There must be a better way, and certainly we should be possible for us to look at what the user is doing, day to day when they are interacting with the software? And could we possibly predict what the user might do with a similar image when it comes? And that's really that's the kind of the basic kernel of this. Is if we put a new image in, the user will do a certain series of actions. Are those actions repetitive? Instinctively, I thought, yeah, they are repetitive. It's pretty much the same. In each software, it will be the same thing. You know, you open the image, you apply some filters, you enhance the image somehow, then you apply your sequence of objects, a sequence of algorithms. This is one example. So in the case of one example of a particular pipeline, there's two pipelines here, but I'll just talk about one example here. With partition, these are the parameters that a particular software, in this case is Avia, requires the user to set. It will ask, what is the soma size? And the user will put some soma size. Do you want to use partition or not? Then do you want to do some intensity threshold subtraction or not? And so on and so forth. So it asks you for the diameter of the objects, blah, blah, blah. It will do all of that. And the user is asked to input those parameters in, and they do. And then you get a good result. Or you get a preview, which then you can iterate until you get a good result. So that's a non-machine learning approach to doing image segmentation, in this case for neurons. It is what I would say the vast majority of the life science community still does today. And it's repetitive. I could plug in here an ImageJ plugin. It would be similar. It would be the same, but it would be a similar problem. And so this, I just quickly calculated here, gives us something like 200 quadrillion possible parameter combinations. It's a ridiculous amount of combinations. Of course, a biologist will know roughly what the size of the soma is, so it can quickly get to a reasonable answer. But still, in the biologist's head, the reality is we're searching through something like 200 quadrillion possible combinations. That's crazy, but we do it very well. So universities work, and it's good to go to university to learn biology so you know what the neuron size should be. But just for scale, the night sky has something like 10,000 visible stars. The human brain has something like 86 billion neurons. Our body is made of something like 37 trillion cells, and we're getting there, but it's something, the number of synapses in the brain is close to 1 quadrillion. We're talking about 200 quadrillion possible scenarios. So we're getting into that ballpark, the kind of options that a human needs to go through in an intelligent way in order to get to the right combination of parameters to do image segmentation using non-machine learning approach, something like 200 quadrillion for one recipe, for one pipeline. And that's similar to the total number of synapses in the brain. It's a crazy exercise that we do in our brain. And currently, to my knowledge, there is no software that will actually grab that user information and try to turn it into predictions. So that's what we tried to do. So what are we doing? We created a system which currently is deployed in Avia that looks at all of the telemetry that we can possibly get. And actually, that was quite a bit of work to figure out what we could get and put basically little sensors on every single part of Avia. And so we collect things like hardware specifications, what the user does within the app, and yeah, as much as we can. We also grab information about the image itself, you know, all the metadata that we can get. And we grab also a representation of the image itself. So we processed an image to create a 1D array. It's basically an image fingerprint, encoded image fingerprint. It's not possible to reconstruct it back to the original image, but it is a good representation. And then we try to teach an AI to learn the relationship between that 1D array and the telemetry data. I should mention at this point, all of this only happens if the user explicitly turns on a flag that says, I want to allow this to happen. Otherwise, it's turned off. If you install the software and you use it, it's turned off forever. It never turns on. You have to go explicitly to a particular place in the settings and turn it on. And there's a lot of documentation explaining exactly what we capture, how we use it, and all of that, as you can expect. But I just wanted to mention that at this point. So we basically put a system in place that can gather all this user information as well as image information. And this is what it actually looks like. We take raw images in, the user puts a raw image in, we convert this using a deep learning model to images that look a little bit like this. It's really a 1D array that we convert the raw images into a 1D array that is unique and anonymized at that point. Each one of these 1D arrays, which looks like a barcode, is matched to the exact user information, user meaning user behavior, not the user name and all of that, just the user information, as well as which parameters they put in the recipe, the image analysis recipe. So the soma size, the intensity, the thresholds, all of those parameters are matched to the specific image that they are analyzing. So then we have pairs of how did the user analyze the image and a fingerprint of the image. We then train a monster model. That monster model then can, given a new fingerprint image, a new fingerprint or a new image that we convert to a fingerprint, given a new fingerprint of an image, it can predict all of these parameters needed to get a good result for that new image. So that's the concept. That's what we have implemented currently. Now some results. I think, yeah, this next one has, yeah. So this is the picture I showed you at the beginning. We now have applied this concept to a big group of images, of course, to see how much can we actually save. And we went from, yeah, on total, it went from like 300, would have been 300 man years or human years to about 80. So it's a really significant saving if we could use that system all the time. And I'll give you a little bit more details next. We did apply it also to non-neuron data. So now we have two pipelines that can do this. One is for neuron analysis, and the other one is for 3D objects, potatoes, basically cells. So we've done that, and you can see here the raw data. Oops, go back here. The raw data, what the human expert would have done, and then what the autonomous AI can do. And you can see, if you look closely, it's not perfect. So this one, for example, is over partitioned. It's pretty well segmented. You get the same kind of mask, but it's over partitioned. This one is, I would say, more sensitive than what the human did, but also the human has a particular interest. So the human, in this case, probably set a really high threshold, whereas the AI is like, oh, I want to detect all of the cells in there, all of the dividing cells, which were not so visible there. You can see there's actually quite a lot of things up here, and the AI decided to detect all of that. But generally, we get pretty good results. They are not always exactly what you would expect, because the AI is a little bit, taking a decision on its own. So summary of a lot of results. We analyzed, I think now, more than 100 different images, 3D images with various dimensions. And on average, a human took something like 12.6 minutes to set the parameters, look at the preview, and iterate through those parameters with the expert knowledge, of course, of a biologist and an image analyst. And is iterating, so manually iterating. Using ATL on its own, so out of the box, no edits, let's say. it takes just 0.2 minutes. Basically, you apply a pre-trained deep learning model on that data set and you get a result. If you do some edits to get to the same result as the human would have taken in the first place, then it takes a little bit longer because, of course, it takes at least one more loop of preview and process. So that's why it's a little bit longer. But still, it's a huge saving compared to the human starting with the raw image and iterating through the parameters on his own. So for now, what we've achieved is a saving of something like 70% of time. Instead of letting the human do all the iterations on his own, we just provide with a preview, which is pretty solid, and the human needs to maybe adjust one parameter or two parameters out of the many that are available. So I'm getting to the very, very end. This is just to remind myself to say that this is definitely opt-in. This is some examples of where you would turn on. So at the start, when you install the software, you get these options. And by default, it's off. You can see here, it's off. Everything is off. If you turn this one on, then it asks if you want to turn the other ones on. There's also a web page that explains everything we capture. It's anonymized anyway, but it explains how we capture it. And then in the app, you can turn things on and off as well. Okay, so final slide, just to summarize the concept. We are looking at how users analyze their images using non-machine learning image analysis recipe. So that's this recipe. And we're looking, how does the user use this recipe to get from a raw data set to a segmented data set? We're then using those parameters that they set. We call that telemetry. And we train models that we've created. We train a model that takes the raw image, converted into a 1D array kind of fingerprint of the image, plus the telemetry. We train those models and we deploy that back to all the users so they can benefit from essentially the usage data of all the other users. So this is really a community effort. It's a passive learning. The user doesn't need to do anything special, just needs to use the software. We gather the information, train the model, and deploy it back. Deploy it back into the software so that people can benefit from it. So that's where we are. We've managed to do this for neurons and for 3D objects. We're looking into time-lapse data. So that has another challenge because the time part, and also relational data is not done yet. But that's relatively easy. It's just not done, not enough time yet. But we've done these two, and we're working on the other two. One other thing that I wanted to mention is if we can predict how to analyze an image and what kind of objects you have in an image, we already have tools in Avia to phenotype objects into classes, object classification, basically. So if we can detect, imagine you turn on a microscope, you capture an image, and you have this system on, it means it will detect the objects, and Avia can also classify the objects. What would be also very cool now is to say, okay, microscope, only capture images that are of a particular phenotype, a particular cell event. And that's what this kind of technology also enables us. And that's also what my team, the data and analysis team at Leica is very interested in. And that's part of the future for sure. So thank you. I hope I was on time, not too far on time. Really appreciate the opportunity, and I'm happy to take any questions.